Now next up, we have enzyme terminology. And before we talk about a bunch of specific enzymes, we'll need to learn a little terminology. Now the name and the function of an enzyme are related, and if you understand this nomenclature, you can figure out the basic function of an enzyme. Now we're going to briefly cover some important enzyme categories. We'll start off with our first category here, the kinases. Now these enzymes transfer a high-energy phosphate group from ATP to a target substrate. An example of a kinase is phosphofructokinase, as you can see here. Now you can find phosphofructokinase in the glycolysis pathway, where glucose is broken down to create ATP. And here, phosphofructokinase phosphorylates or adds another phosphate to fructose 6-phosphate in glycolysis. And this will form fructose 1,6-bisphosphate, as you can see here. And just to recap that, fructose 6-phosphate is acted on by phosphofructokinase, which takes a phosphate from ATP and sticks it on fructose 6-phosphate to form fructose 1,6-bisphosphate. The thing to remember here is that kinases transfer a high-energy phosphate group from ATP to a target substrate. Now next up, we have the phosphorylases. And what do you think they do? Well, the phosphorylase enzymes are similar to kinases, meaning that they add a high-energy phosphate group to a substrate. The difference here is that this enzyme does it without using ATP. An example of a phosphorylase would be glycogen phosphorylase, which is found in the glycogenolysis pathway. Now, glycogenolysis is where you take a large glycogen chain and break off free glucose molecules. And here, glycogen phosphorylase grabs a free phosphate and cleaves the 1,4-glycosidic bond in glycogen to produce glucose 1-phosphate. So what do you need to remember here about the phosphorylases? There you go. They add a high-energy phosphate group without the use of ATP. Next up, we have the phosphatase enzymes. And these are the opposite of kinase and phosphorylase enzymes. Phosphatases, like fructose 1,6-bisphosphatase, remove a phosphate group from a substrate. And here, fructose 1,6-bisphosphatase converts fructose 1,6-bisphosphate to fructose 6-phosphate. And this reaction can be found in gluconeogenesis. And in this process, we saw a substrate with 2 phosphates attached. And after enzymatic removal, the product only has 1 phosphate. So once again, the important point here is that phosphatases cleave off phosphate groups. Now next up, we have the dehydrogenases. And these catalyze oxidation-reduction reactions. And while we're on it, do you happen to remember what a redox reaction is? There you go, it's the exact same thing. It's an oxidation-reduction reaction. And this means that hydrogen and its corresponding electrons are removed from one substrate and added to another. An important example of this is pyruvate dehydrogenase, which converts pyruvate to acetyl-CoA, as you can see here. Also here, you'll see that pyruvate dehydrogenase is moving electrons and protons to form NADH, which will then be used later. The big thing that you need to remember here is that dehydrogenases catalyze oxidation-reduction reactions. And this just means that hydrogen and its corresponding electrons are removed from one substrate and added to another. Now next up, we have the carboxylase enzymes. And these move carbon dioxide groups onto a substrate, as you can see here. A great example of a carboxylase enzyme is pyruvate carboxylase. And here, a pyruvate molecule has a CO2 group added with the help of pyruvate carboxylase. And this forms oxaloacetate, which is a molecule with 2 CO2 groups. Now the carboxylase enzymes need a little help. Therefore, they have a cofactor. And do you know which one this is? There you go, it's biotin, or vitamin B7. So once again, the important point to remember here is that carboxylase enzymes move carbon dioxide groups onto a substrate, and they often need a cofactor, which is biotin, or vitamin B7. Now what do you think you'd call an enzyme that actually removes CO2? There you go, that'd be a decarboxylase enzyme. And as we just stated, the decarboxylase enzymes remove CO2 groups. Now what about the hydroxylases? Well, these are enzymes which end up adding hydroxyl groups, as you can see here. Now how about the mutases? Well, the mutases are enzymes that move functional groups around a molecule. An example of a mutase would be methylmalonyl-CoA mutase. And just to review these one more time, decarboxylase enzymes remove CO2, and hydroxylase enzymes add a hydroxyl group. And mutase enzymes move a functional group from one spot to another on the same molecule. Now next up, we have the synthetases as well as the synthase enzymes. And these work to join molecules together. The only difference here is that one uses ATP while the other does not. Now can you guess which one actually does use ATP? There you go, that's going to be the synthetases. And they use ATP or GTP, while the synthases do not. Now an example of a synthetase is carbonyl phosphate synthetase, an enzyme which converts ammonia, NH3, and CO2 with the help of 2 ATP to carbonyl phosphate, as you can see here. Now an example of a synthase enzyme is citrate synthase, which converts oxaloacetate plus acetyl-CoA to citrate, as you can see here. So once again, the big point to remember for synthetases and synthases are that they're enzymes that join molecules together, and that the synthetases use ATP or GTP, while the synthases do not. Alright, well that'll wrap up enzyme terminology. See you in the next video.